Okay, welcome everyone to this ERGA seminar. I see many people is connecting now to the YouTube channel. And without further ado, I will introduce the speaker of today. He's Schikora Tamarit, and he's a postdoctoral researcher in the Barcelona Supercomputing Center and also at the Institute of Research in Biomedicine in Barcelona. And he's going to talk about PerSVade, which is a tool for personalized structural variation detection, which we hope is going to be useful also for you when working with non-model organisms. So whenever you want, Miki, you can start.
Okay, thank you, Tony, for the introduction. And I want to thank the organizers for letting me talk here about these pipeline that I developed during my PhD. Just to clarify, the seminar objectives are going to be speaking about the pipeline itself for 20 minutes, and then I'm going to provide a little tutorial on how to install and also use PerSVade. And at the end, we'll have some open discussion that everyone can share their doubts on kind of this 20 minutes again. So I'm going to start with the first part, which is about this pipeline overview. Which is basically what we report in this paper from a couple of years ago. I want to start by stating why we believe that we need a species-tailored pipeline to call structural variants. And this goes to these this very, very broad thing, which is a genetic variation that is related to many fields. For example, you may be studying how specific genetic variants contribute to human diseases or to antimicrobial resistance. For instance, in my PhD, I was studying these processes. Or maybe you're interested in how they shape natural diversity, or how they, or maybe you want to use genetic variants to define strains of some pathogens like SARS-CoV-2, right? All of these things require the technical part of studying and identifying these genetic variants from some sort of sequencing data. Now, there's different types of genetic variants. So most of you probably are thinking about these single nucleotide polymorphisms, which are these very simple variants where you just have one change in one position of the genome. These variants are very easy to detect, let's say, fairly easy to detect when you have short read sequencing data, which is what we have most of the time, because it's easy to map the reads to the reference genome and pinpoint the location where the variant is. However, there's these other types of variants that have been gaining attraction in the last, I would say, 10 to 15 years, maybe, where you have these are structural variants where you have like a change in the genome that like large changes that generate things like deletions, duplications, inversions, which are much more difficult to detect from short read sequencing data because the mapping aspect is not as simple. So which actually explains why they have been less studied, because the technical aspect of detecting them is quite more complicated. Still, we know that they are driving very important phenotypes. Here I'm putting just 3 examples of that show that structural variants are related to, for example, Parkinson's disease, cancer, and also drug resistance in fungi, which is more related to my specific field of expertise. However, despite that we know these variants can be very relevant, they have been much less studied than other variant types just because of these difficult detection for short reads. And I want to stress that we strongly believe that it's relevant to be able to detect these structural variants from short reads. Many of you may be thinking, why don't we use long reads, which are more suited for the task, right? So I think there's many reasons why it's interesting to still be able to do this from structural variants. The first is that short read sequencing is cheaper, which becomes relevant when you have large projects of sequencing many individuals, which means that if we have a pipeline that can detect structural variants from short read sequencing, we can have more cost-effective approaches to genomics. Short read sequencing is more widely used, for example, in clinical, but also, I'd say, academic settings. So if we have a pipeline to call structural variants based on them, we could reanalyze a lot of data, so gain lots of new knowledge about these important variants. And then finally, the short reads are usually better for SNP calling. So if we have a pipeline where we also can find structural variants, it means that with a single library, we can detect many different variant types. So this is why we believe it's really important to have tools to detect these variants. And I want to provide an overview of what these tools are. So I would say in the past 5 to 10 years, there have been developments in the tool in the tools. So where there are some tools that take short reads and based on things like de novo assembly or evidence from split reads, they can infer the regions of the genome that have structural variants. These tools have been tested on the problem is that they have been tested on a few species, typically just human. So when you want to use these tools on any other species of interest, for example, I wanted to use these tools for fungal pathogens, you come with these two very relevant questions, which is how do you choose the filtering parameters and how so will these tools work in my genome? So what is the accuracy for my species that maybe has a very different content of repeats, or maybe the data set has a very different coverage than what's used for the human data sets, right? And let me stress this issue of filtering parameters. So basically, this is what you get when you have one of these tools. The output that you get is like a list of different structural variants, and you get some statistics about them. So you typically want to specify some, for example, minimum number of reads covering a variant, the minimum quality. Maybe you want to discard some reads that are overlapping discard variants that are overlapping some complicated repeats that generate false positives. So setting these thresholds is what's called the filtering parameters. And we know that setting them correctly is very essential for having accurate calling of the structural variants. But again, we don't really know how to set these for many species, which is what we try to address with our pipeline, this PerSVade pipeline. I'm going to be talking about how we built the pipeline and how we tested it. So it's called personalized structural variation detection. The idea is you will call structural variants in a way that is personalized for your species of interest. And it has these two main modules. The first is one to take short reads in a reference genome and map them. And then the second module basically takes these reads and identifies the structural variants. And this is done in two parts. The first is inferring a list of breakpoints, which is a list of regions of the genome that are rearranged. And the second is taking this list of breakpoints and summarizing them into the actual structural variants, which are these, basically some simple SVs and also more complex like translocations. So these are like the variants that we find with the pipeline. Now, this would be like a standard pipeline. The main and most interesting module is it's related to this optimization of filtering parameters. Basically, what it does is it does simulations of structural variants, and it uses them to learn which are the best filtering parameters for the simulations. And we do the simulations in a way that resemble the input, in the sense that they are based on the same reference genome and they have the same technical properties. For example, the same coverage, the same read length, right? So with these, you basically get the best possible structural variant parameters that are adapted to your species of interest. You get a report of the accuracy on these simulations, which is very useful to understand whether you can use that pipeline on your sequencing data. The typical thing is, I have a very low coverage. Can I call structural variants? So this is a way to address this. And in general, with these three modules, we believe that we provide a very simple way to, in very few commands, get the filtered structural variants directly from the reads, as compared to the previous analysis. Now, we try to test PerSVade and test this concept. In the paper, we do many testings. I'm going to just talk about the testing we did on simulated structural variants. So basically, we analyze these three. So we analyze 6 species. These are 3 eukaryotes of different clades, and so six eukaryotes from different clades, and 3 short read data sets for each of them. So basically, we had these 2 questions. What is the structural variant calling accuracy? And how relevant is this parameter optimization for each of the samples? Because maybe we can optimize for one and it's going to be enough, and we will always get a universal set of optimal parameters. That's what we try to analyze. So in terms of accuracy, what we look is at this F value, which is this harmonic mean between the precision and the recall, which can only be high if both things are high, right? Actually, PerSVade is trying to maximize this F value to get kind of a balance between the two to get the most accurate parameters. And I'm going to show these results. For example, this is regarding the first question: what is the calling accuracy? This is the results that we have for Candida glabrata. So this is for different types of structural variants, this accuracy value, when we are using either optimized parameters or default parameters that we rationally designed based on previous papers. Basically, we see it's a huge difference, right? So thanks to this optimization of parameters, we really increase a lot the accuracy. We did this for the other species where we see a similar trend. The changes are not as dramatic, but in general, we can say that thanks to PerSVade and this optimization of parameters, we're really improving the variant calling accuracy. Now, we had also this other question: how relevant is this parameter optimization? Maybe we are just finding always the same set of optimal parameters, so we would not really need to do this for every species. So kind of a more specific way to frame this question is to ask, how do the best parameters that are optimized for one sample, how do they work on all the others, right? So we basically run this parameter optimization for 3 data sets for each species. So how do these parameters work when we test them on the simulations of all of the other data sets? And this is the result. Basically, the red is kind of higher accuracy, the blue is lower accuracy. And you can see how there it's kind of, it's not a, we don't find universally good parameters. Very often the parameters that work for one species, they do not work for the others, right? Which means, and one trend that we do find is that the parameters that work for the samples of one species tend to work for the other samples of the same species, but not for the others. So in general, this shows that we need parameter optimization for each species to get these accurate structural variant calls, which shows the relevance of the approach that we're trying to do here. I have to say that now I've been talking about, so PerSVade has these different modules. I've been talking about these 3 modules that are related to structural variant calling and the parameter optimization, but there's many other modules related to the calling of small variants, SNPs and indels, the calling of copy number variants based on changes in coverage. There's tools to annotate the variants and also to integrate different types of variants. So you can think about PerSVade like this kind of Swiss knife that can do many different things. The structural variant part is the more scientifically novel, but it also allows to work with all of these different variants at the same time. And we built it in a way that has some reproducible installation, so that hopefully people can be using it and kind of successfully identifying these different types of variants. This is something I'm going to be explaining more in the second part of the talk, which is tutorial. But before I want to explain, I want to mention an example application of PerSVade, so a study where using this tool was very relevant for the conclusions. So basically, this also comes from my PhD research, where we studied how antifungal drug resistance evolves in vitro in Candida glabrata, which is an important human fungal pathogen. So basically, we did this very, very interesting experiment of taking several strains of Candida glabrata and growing them into increasing concentrations of fluconazole and anidulafungin, which are 2 drugs of clinical usage, to get resistant strains. And then we also did, in another part of the experiment, the combination therapy to get multi-drug resistant strains. Then we sequenced 77 strains evolved in these different conditions. So we sequenced the whole genome with short reads. And then we identify different variants, structural variants, SNPs and indels, thanks to PerSVade, that appear during the experiment. And these variants tell us something about the mechanisms of resistance. So in the following minutes, I'm going to be speaking about the structural variant part. And specifically, I'll show you examples of how using these ColSVs, which is this module to find structural variants, how these results of this module compare to a more traditional detection of structural variants that is based on coverage. So let me stress these. So this is how PerSVade identifies structural variants, basically taking identifying breakpoints and then identifying this precise structural variants. The typical way how, at least in this field of fungal pathogens, the typical way how people identify structural variants is by just looking at coverage across different windows of the genome and saying, okay, if I find a region that has higher coverage, it's a duplication. If it has lower coverage, it's a deletion. So I'm going to show examples of why using this kind of approach, that is the core of PerSVade, provides further information versus this one. For example, we find structural variants in this chromosome E that contains the drug target of this ERG11, which is the fluconazole drug target. Basically, this is how these two chromosomes look in the wild type strain. This here you have this gene that we think is important. So we find one strain evolved in fluconazole that has basically a duplication of this region and a kind of a unbalanced translocation and attachment to chromosome J. Now, if we think about, okay, what would have we found if we were just looking at the coverage? So we would see the duplication. We would see a duplication in this region. But thanks to PerSVade, we really understand what's the source of the duplication. So we understand better the mechanism of the structural variant formation. Yet another example, which is in FKS1, we also find some interesting structural variants. This gene encodes the anidolafungin drug target. So this is how it looks in the wild type. We have FKS1 here. There is this strain evolving anidolafungin where you have like this balanced translocation between the two chromosomes, which basically breaks the gene. And this is not changing the coverage. So thanks to PerSVade, we can identify this variant at all and understand that this gene has been disrupted through this translocation. Here is another example from this ERG3 gene that we discovered can be related to anidolafungin resistance. And this is how the gene is in the genome and how it looks in the wild type. Basically, we find a strain evolved in both drugs at the same time that lacks this tiny little part here. Actually, this part is so tiny that with the coverage-based detection, which relies on putting some windows of the genome, we would not find it. So thanks to PerSVade, we can really identify also these very small deletions that can have a very, very, very important impact on the gene because it's at the beginning. Here is another example with CNE1. It's another gene that we find related to anidolafungin resistance. And so here, this is where the gene is. And this is how these two chromosomes look in the wild type, I say reference strain. So this is the strain for which the reference genome was sequenced. The interesting thing there is this other strain that we use in our experiment that has a balanced translocation between chromosome D and L. And so when we evolve this strain in anidolafungin in one instance, we see that this region here is lost. It's kind of a single deletion. So if we were looking at the coverage, basically we would see a deletion here and a deletion here because we always map towards the reference genome. However, thanks to PerSVade, we can understand that actually we had this first a balanced translocation and then a deletion. So we better understand the mechanism of structural variant formation, which I find kind of improves a lot our understanding. Here is a slightly related example, which is we see there are structural variants in chromosome I related to fluconazole resistance. Again, in the wild type reference strain, we have these 3 chromosomes, but there is this other strain that we use in our experiment that has these two balanced translocation. So it has this kind of mix of chromosomes. So there's two strains that were evolved from fluconazole of this one that had a duplication of these kind of mixed chromosome, which is much shorter. So again, if we compare this to what we would see with the coverage-based approach, we would see that there is a duplication here, one here, and one here. So thanks to PerSVade, we can understand it's a single duplication of a whole chromosome. We actually think that the fact that this is a smaller chromosome makes it more likely in this strain to have this kind of duplication because the fitness cost of duplication a small chromosome is kind of high, is much reduced. So thanks to this understanding of the variants, we can also derive some interesting biological and evolutionary insights. And basically, that's all of what I wanted to show you today. I hope I've convinced you that PerSVade can be used to detect accurate to detect structural variants in an accurate way. I've shown you that parameter optimization is likely necessary to get accurate results. We don't find just a single set of universally good parameters, but we need to adapt to each species. With the example of Candida glabrata, I hope I've shown you that this pipeline is useful to do some genomic analysis, especially in non-model organisms. And I've tried to compare the results that we get when looking at structural variant analysis by PerSVade versus a more traditional coverage-based detection. And we see that PerSVade improves these. And basically, if you want to know more about the pipeline, I really recommend you go to our GitHub and also read our paper where we have all of this information. And thanks a lot so for your attention. So now we are going to be, the questions about this topic, we can have this more long discussion at the end. But now I'm going to provide a more in-depth tutorial about how to use the pipeline, specifically like which commands to use. And basically, this is what I'm going to say in this section is a reduced version of what you will find in the wiki in our GitHub repository. I want to say, so this means that you don't need to memorize or kind of write down everything I say. So we will find everything even better explained in the wiki afterwards. So the wiki has these 8 sections. I'm going to start by the part of installation, which is kind of tricky because this is a very big pipeline with many dependencies. So the first type of installation that we support is via Conda environment. So PerSVade is basically uses around 10 Conda environments. So this is how you install them. First, you install, so you download the latest release of PerSVade that you can find here. You uncompress it, and then then I'm going to show you some commands that will create these Conda environments on which PerSVade works. So you have to install Mamba, which is a faster version of Conda. Then you define the name of the environment. Then you create this main environment. Then you activate it. And then you also install some external software that cannot be installed with Conda. And then there's 13 more commands dealing with the installation of all of these different environments. So this is kind of we provide all of the steps, but it's a bit of a tedious installation that may not work all of the time, I have to say. But here you can find more information in our wiki about how to do it. Now, the interesting thing about Conda is that it's very easy to run. So you just say Conda activate the name of the environment, and then you execute this PerSVade script, which is the main wrapper that will launch all of these different modules of PerSVade, right? Now, of course, this kind of installation is tedious and it may not work in some systems. So we also provide a Docker-based installation, which increases a lot the reproducibility as compared to Conda. So to use Docker, so you first have to install it. This depends a lot on the operating system that you are working with. For example, in Mac, I use Docker Desktop, but there's other options. And then you have to install what's called the PerSVade image, which is this one with Docker pull Schikora /PerSVade. This installs the image. So the tag is like the version. You have to go to Docker Hub and see which is the latest version when you want to use the pipeline. I always recommend to use the latest version. So what this creates is this Docker image, which I understand it as like a virtual machine that is running inside your host computer, and that contains all of the pipeline code and all of the software environment. So it's a really nice way that you can run PerSVade the same way as I do, because it's in this kind of isolated, virtualized environment. Now, this is how you run this Docker image. You say Docker run, you provide the name of the image, and then you execute this command that is basically everything that goes after here is executed inside of the image. So with all of the environment of the image. So this would be the command that we would use to print the help message of PerSVade. Now, there is one important caveat here is that, so the Docker image doesn't know any files, so it doesn't have access to any of the files of the host computer. So for example, if you want to provide inputs to the pipeline, you have to what's called mount some folders, and this is what I'm showing here. So basically what you do is you specify with this -v, you say this is the folder in the host computer that contains some reads, for example, that we want to analyze, and I link them to this folder inside of the Docker image. And then you, in the PerSVade command, you will be using the path of the Docker image. And this seems easy enough, but if you have many inputs, these Docker commands can get very messy because you have to specify all of these mounting points. So that's, and Docker has also other problems. So we also provide a way to use our Docker image through Singularity, which is another software to run images and containers. So to use it, you first have to install Singularity, and then you build the Singularity image, which is based on the PerSVade Docker image. So this works like this. So you say Singularity build, and then the name of the Docker of the Singularity image, which is actually a file in this case, And then the name of the PerSVade Docker image, which is in Docker Hub. Again, the tag, you will find it in Docker Hub. I would say use the latest tag. So what this does is it creates this zip file, which is it's a file that contains all of the code and all of the environment to run PerSVade. It's really nice because then you have a file that you can really share with people and reproduce. There's two important paths if you want to use it that you will want to remember. The first is this path here contains the main wrapper script of PerSVade that will be launching all of these commands. The second is this the path that contains all of the conda environments that are used by the pipeline. And you're going to you'll need to use both of these paths for this. So this is how you run this Singularity image. It's very similar to Docker. You say Singularity exec, the name of the Singularity image, and then followed by commands that will be executed inside of the image. So this is a Singularity command to execute the main wrapper script. It's a bit more wordy, so I'm going to try to digest it for you. So first, this is the command that is run inside of the image. So it has, it's basically a bash command that starts here and ends here. And it has two parts. First, the activation of the PerSVade environment. And second, the actual running of the PerSVade script. So that's how you would execute it. Now, I have to say, the interesting thing about Singularity is that it has access to, by default, you don't have to mount all of these folders as with Docker. You just have access to many folders like the current directory, the home, and all of these other folders. So you can use the path for most folders. You can use the path as if it was your host machine. But you can also mount other folders with this -b, which works the same as for Docker. And so basically, the point of this, everything I've been saying, is that we provide these 3 different ways that you can install the pipeline. And I'm going to try and present the pros and cons of each of them. I am spending some time here because from users, I've seen that the installation of the pipeline is one of the most challenging times because the Docker and Singularity sometimes are a bit scary to use. So for the conda installation, it's nice because you don't need to know any conda in Singularity, so it's maybe more straightforward. But it's quite more complicated. So you have to execute all of these different commands, create all of these different environments. It's restricted to Linux, and it's likely not 100% reproducible because conda relies on some system libraries that probably will be different across different computers. Now, Docker is really a nice fix for this because it enables a reproducible, very simple to install, very simple to run in any operating system. But it has some caveats. So it requires root permissions. That's a huge problem if you want to use high performance computing machines because you won't be able to use Docker. The user needs to specify all of these mounting points. So the inputs and outputs, you will have to specify all of these folders, which can get a bit messy. And in general, you need to know a bit of Docker to use it. So we provide all of these commands in the wiki. So you don't need to be an expert in Docker, but it's a bit more complex in the sense than conda, for example. And then we also provide this Singularity, which is this way of running a Docker image through a slightly different system. So it's nice because it enables reproducibility in Linux and Mac. It has a much simplified read and write system because it already mounts by default several folders as compared to Docker. It can be run in systems with no root permissions, and this is nice because in high performance computing, you will only be able to use the Singularity images. And in general, the way how Singularity works, that it generates this zip file that contains the Singularity image is really nice because you can transfer it across machines. You can send it to some collaborators. This is the exact code that I use. It's kind of a file, right? That's very nice. However, it has some caveats. The first, it's not available for Windows, and you will need to, as far as I know of, and you will need to know a bit of Singularity, which is can be a bit of, you have seen how the Singularity command is a bit wordy, so you'll need to be using this kind of a bit more complex syntax. So due to all of these pros and cons, I would generally recommend using the Singularity image unless you run into some issues. But that's what I'm going to be showing several commands on how to use it, and I'm going to be always showing the one of these images. Now, in the next section, let me get some orders, sorry. So I'm gonna provide an overview about how to use PerSVade and understand a bit what the outputs of the pipeline are. So which is basically following these 4 sections of the PerSVade wiki, where you can find everything that I will be saying. Now, let's say that you want to align some reads to the reference genome with this align reads module. Basically, this is the command that you would be using. Let me digest it for you. So first, you execute the singularity image, and then after this, you will be executing inside of the image this bash command that starts here and ends here. And this bash command has 2 main things. The first is the activation of the PerSVade environment. So basically, you do the source of the conda profile that is inside of the singularity image, and then you activate the PerSVade environment. This is the one that you will need to be using. And then in a second step, you run the PerSVade command. So you would say Python, the path to the PerSVade script inside of the image, followed by the name of the module, in this case, align reads, and then followed by the arguments of the align reads module, which is the reference genome, the output directory, and then forward and reverse reads. And with this command, you would get, basically, it would run and then would output all of the map reads. So these are the outputs of the pipeline. You have the sorted reads in BAM format. Also the index of the BAM, and then also a file that has the coverage per windows, which is useful for getting some stats about how the mapping went. Now, there's also these other two files that are output by the pipeline, which is one is all CMDs, which this contains all of the individual commands that have been run inside of the module, which is very useful if you want to know how the mapper, exactly how we codify the mapper to run, or the indexing of the genome or anything. Here you can find it. And then you have also this PerSVade finish file, which is only created when the module runs successfully till the end. So this is very useful to integrate these PerSVade modules into whatever workflow you're working with. Now, this is one module, but PerSVade has many other modules. This is a picture that you will also find in GitHub that is very useful to understand which modules to use and what is input of what. So you have to kind of, depending on the task that you want to do, you will want to use one or the other. So I'm going to show you how you access them from the code perspective. So basically, you execute this PerSVade script, and then you get this help message that basically shows the available modules. And then this kind of tells you how the usage works. You basically say PerSVade, the name of the module, and then the arguments of the module. And you can type PerSVade, the name of the module, -h, to get information about each of them. So, for example, if we type PerSVade align reads -h, we get something like this. First, all of the arguments that each of the modules has. I mean, this is a complex pipeline, so it will have like lots of arguments. Also, a kind of a message explaining what this module is doing, and then an in-depth explanation of all of these arguments, what they are for. So this is how you learn about each of these modules and how to use them by reading and going through all of these steps of getting these error messages. So in the following, in the remaining part of the seminar, I'm going to show you how you execute each of these different modules, as well as what outputs they give, so that you get a grasp of what the pipeline can do, and maybe you find some applications for your own research. First of all, we have this module. This is a very simple one that trims reads with Trimatic and then runs FastQC on them to perform the quality control aspect. Basically, this is how you run it. You say Python PerSVade, the path of the PerSVade script, and then followed by the name of the module and then the arguments. This is going to be the same for all of the arguments. So here you basically specify the forward and reverse reads and an output directory. Now, note that you have to embed this command inside of this conda activate that we were saying before. So this is just the part of executing PerSVade. And this outputs the trimmed reads and also a FastQC report that shows you how well the trimming went. Let's say that it's very important that you don't just use the reads right away, that you check these FastQC reports to see if there's some issues in the sequencing. So that's actually in the pipeline module, there's this I here that says you should check the reads before providing them to the other module. Right? So now the second part would be these module line reads that we have been touched on. And now I'm going to speak about a module that is called infer repeats. Basically, this is a module that is relevant because for other modules. So let's say this module takes the reference genome and then infers the repeats, the repetitive elements inside of it by using repeat modeler and repeat masker. And these repeats will be used in other modules. So that's why I explained them at the beginning. This is how you run it. Basically, just provide the reference genome and the output. And it outputs several things. The most important one is this combined repeats.tab that contains all of the repeats that you will be providing to other modules. For example, this optimized parameters module that generates these best filtering parameters. You basically run it like this. It's a bit wordy. I'm going to try to ingest it for you. So you basically provide the structure, the sorted BAM. These are the aligned reads from which we will be reading the, so this module will be reading the coverage, the insert size, the length. You also provide the mitochondrial chromosome because some operations are different for the genomic DNA and the mitochondrial DNA. You provide the reads that you have got with this infer repeats module. And then you have to specify also the regions where you want to do these simulations of structural variants. By default, it's randomly placed, but it could be anything else. And then you also have to say the ploidy that you are working on. And the outputs, you have many outputs. The most important ones are, so you get these optimized parameters.json. This is a file that has the considered best parameters that you can use then for other modules. And then it has also several files that will help you understand with some plots and also some files how well does the pipeline work. So how accurate is it on these simulations. The next step is you take these best parameters and you provide them to CallSVs, which is the main module of PerSVade, which basically it's run like this. And it has basically most of these arguments I have already mentioned before. So there's one that is different, which is you basically provide these SV column parameters. This would be the path to this JSON file. It's really nice because also the JSON file, you can edit it. So if you can also customize however you want. But in principle, you would obtain this file by the optimized parameters module. And this module outputs several things. The six first are the individual types. So all of the structural variant types. And then there's also several intermediate files that are the outputs of GridSS and Clove, which are the programs that this module is based on, that maybe you want also to use. So this would be all of the structural variant, let's say. Then we also have this call CNVs module that basically performs this more traditional coverage-based calling of copy number variants, or it's a type of structural variant. Basically, first it runs, it uses mostdef and Python to measure coverage per windows of the genome, and then it also applies some correction to this coverage to remove the biases that are imposed by things like GC content, mappability, and the distance to the telomere. And then it uses this module uses this corrected coverage to infer with 3 different programs duplications and deletions, which are these regions. This is how you run it. Basically, you say, I'm going to outline the important arguments. You have to specify the ploidy that you're working on, the calling algorithm. So we generally recommend using HMM copy and anneal finder, which are the most accurate. You have to specify the window size in which you do the calling. This will depend a lot on the genome size, actually, and the resources that you have. And then finally, the reference genome. And with this, you get several files. The important one is this one, the final CNV calling. This is a tab file with all of the copy number variants. And then there's also a couple of plots that are interactive HTML files that will be useful to understand how how this process of correcting the coverage and how this copy number variation calling went, which is very useful because these tools are not very accurate very often. So it's really nice to be able to see how they work. Then we also have this module is also kind of an innovative part of the pipeline. So when you have when you run these 2 programs, you get many different files. So this is a module to integrate all of the copy number variants and structural variants into a single VCF file. Basically, you run it like that. The important thing here is that you have to provide the output of CallSVs and the output of call CNVs. And then this generates a single file, which is the integration of all of the variants in a way that is non-redundant. And then this is kind of a complex file that we provide a lot of documentation on how to understand how it's encoded. There is a frequently asked question to this, so in case you're wondering, this is a place to check it. Then the interesting thing about this, because this is like a typical thing. So I call all of these variants. What do I do with them? How do I work with them and do some downstream analysis? So a typical downstream analysis would be which genes are affected by these variants. And we provide these annotate as these that will do this. Basically, what it does is it uses the ensemble variant effect predictor on this VCF of structural variants. So this is how you run it. You provide the GFF file with the annotations with all of these genes. Then you have to specify the translation codes that could be very different for different species. For the mitochondrial DNA and also for the genomic DNA, you can find in NCBI these different translation codes. And then you provide the VCF with the structural variants that are merged. And with this, you get a file that has all of the annotated variants. Then there's also a program to call small variants based on three different, so with small variants, I mean SNPs and indels, but based on three different programs. Basically, this is the command. You have to specify the colors, so the different algorithms, the minimum allele frequency that you want for your variants, and also the minimum coverage for positions to be considered for the column. And you get several outputs. So most of them are like all of the raw variants that all of the different programs give. And then there's also some filtered variants based on the filter that you specify. And then finally, so there's also a module to annotate these small variants to see how they are linked to genes, which works very similar to the one of the structural variants. You just provide the VCF and like the GFF, and you get the annotated variants, this mapping between variants and genes. And then finally, a very simple one, but that can be useful, is it's a module to calculate coverage per gene. You basically just provide a reference genome and the GFF and the sorted BAM, and you get a file that has all of these genes and regions with the coverage. It's just like for a quality control thing, could be very useful. So there, I want to end with these general tips and tricks. So all of these modules generate this file that is very useful. So it indicates a successful run. This is something you want to use for integrating these modules in your own workflows. PerSVade is done in a way that if the run breaks for some reason and you rerun it with the same arguments, it will not repeat any steps. So it's kind of a way like workflow managers do. The philosophy of these modules is that they have several mandatory arguments that ensure that you will need to understand what's going on. So it's not going to be all default arguments. So there's going to be some things that you will need to understand what's going on to set them. And I think this is a good thing so that the pipeline is not just doing lots of analysis that you don't want them to do. And there's these three arguments that are relevant for setting the resources to allocate threads and also the memory. And you can check the wiki on how they use, but they are very useful for large data sets. We have this very extensive frequently asked questions section in our wiki, which I encourage you, if you use the pipeline, that you check it, because there's like lots of tips and tricks and how to work with it. And in general, if you're using the pipeline and you run into problems, I'd recommend you raise this a GitHub issue in the same repository to ask for help, because this is better than an email, because if you send an email to me, we won't be able to, like, other people won't be able to benefit from our discussion for solving their own problems. So this is, I think, the preferred way to ask for help. And this is pretty much everything I wanted to say today. Regarding the tutorial, I hope I've convinced you that PerSVade provides this flexible and simplified installation options to obtain all of these variants. So it can work with all of these different types of variants, SNPs, indels, structural variants, and copy number variants. We provide tools to filter them, to annotate them. So it's kind of a toolkit that can do all of these things. And we have designed it with this modular structure that enables running only the parts you need. For example, you may only want to run SNP calling or annotation or repeats, and you can do this fairly easy. And again, if you want to know more, I really encourage you to check out our GitHub and our paper, where you will be able to see, to refresh everything I've been saying today and also find more information. And I want to thank you a lot for your attention, and also all of my lab mates that were very, very helpful with the development of this pipeline. So this is our lab picture from the last retreat. And I think now probably we can move to the part of the open discussion. In case there are some questions, I'll be really happy to answer them and discuss them. Thank you.
Okay, thanks a lot, Miki. It was a very clear presentation. And now we have some questions in the chat. I don't know if you would prefer to read them yourself or you want me to get them to you. So I can read them to you. So
yeah.
So the first question is by
Yeah.
Chiara Albertini. So
Yeah.
she's asking in the heat map where you show the optimal parameters for one species, how they work for other species. How do you explain that in Drosophila, the parameters seem not to be optimized for other Drosophila species?
Yeah, that's a very good point. So basically the Drosophila, so the way how we selected these different data sets was based on finding the three latest ones. So we picked randomly 3 data sets from that were matching some criteria, but with Drosophila we picked one that had much lower coverage than the others. So in the ones that have lower coverage, we see that also the accuracy is lower, which is what we would expect. And this was not the case for other species. I hope these so yeah, and actually I think this shows that of course there is some limitations with a much lower coverage. You cannot find you cannot have very accurate detection. But yeah, so there's more diversity in this. That's what explains it.
Yeah, thank you. So Gabriel Mochales is asking, well, he says really interesting software. What should be the minimum coverage per sample to work with it? And are the results comparable between samples with different coverage? And he thanks in advance.
Very, very good question. So this is actually something I was very surprised by. So we would expect with very high coverage or much like to have better accuracy. But what my impression is that having a coverage of around 30 is like ideal. It's like what would be like high coverage in humans. So for example, we see that in Candida glabrata, so it's one of these fungal pathogens where we have very high coverage of around 300. The reason why we believe that well, we actually tested this also in the paper. So having 300 coverage, what it makes is that some parts of the variant detection doesn't work very well. So when we downsample to 30, it works better. So I would say 30 is like the gold standard. Actually, that's what these tools were designed for. So if you have very different coverage in one population, for example, I would recommend you use so you either downsample if you have a lot very high, or you optimize parameters for each of the coverages. So it really impacts. Now, as for medium coverage, I think the Drosophila one was like 5x, and it was not like you would see a drop. But for 10x in some species, we saw that we could, at least in the simulations, find quite accurate. But again, you can check these with the pipeline. That's the point. You can run this optimization yourself with your specific data set, and you can find out.
Okay, thanks. Then another questions by Yota or Obeta. I don't know if I pronounce well. If I have 2 alternative reference genomes, one is larger than the other one, which I should use? So regarding the reference genome,
Okay,
if you have 2 alternatives, one for the same species, I guess.
I would say probably you want to use the one that is closer. I mean, first the one that has like, so there's 2 things. One is how close is your sample that you're analyzing to this reference genome. Probably the closer I would say is better. But then maybe the other one, it's more far away, but then it has some kind of the assembly is worse. So having the best possible assembly is also an important thing. So I don't think it's a thing about size. So, but it's more about which genome is better and whether it's closer or not. I hope this solves this specific issue.
Yes. So the next question by Malin Hasselgren, is this pipeline also suitable for long reads?
No, I'm sorry, but no. So the whole it's like we get this that we should be doing this for long reads. So all of the filtering parameters, the intrinsic, so the software, the underlying software are for short reads. So it would not. I don't think it would make sense to use it the same way for long reads. But maybe in the future we will be working on doing something similar for long reads. But Yeah.
Okay, now Teresa Manusaki is asking, how does the software compare with other variant calling software?
Yeah. For these, this is something we did not do because so the colors that we're using, these grid SS and Clove, these ones have already been benchmarked against other types of software. And so we picked them because people found out in other benchmarks that they were among the top ones, right? So the thing is, PerSVade is not really a new software; it's just the optimization of the parameters. So the question of whether it's better than another software, it really depends on which parameters you're using. So that's why we didn't compare it specifically.
Yeah. Then Maria Madrid has a couple of questions. The first one is this pipeline able to detect very large structural variants, for example, a very large inversion that overshoots the length of short reads?
Yeah, for sure. I mean, basically, that's the main point. So with you basically the way how these structural variants are detected is based on split reads, also discordantly aligned pairs, like you have one pair here and the other is somewhere else. That's an evidence for the structural variants, and also de novo assembly of these kind of complicated regions. So it integrates these three types of information. So it should be able to handle any also interchromosomal things and like any type of structural variant in principle. So
yeah, and the other question is related. Maybe does it work only with paired short reads, and also how it performs with draft reference genomes?
Okay, the question of paired reads, yes, you need paired reads because I don't think so. For short reads, I think the whole aspect of de novo assembly and as I was saying, the discordantly paired reads, this is like what the software relies on, so you need them. And I was not seeing the second one, the draft.
How it performs with draft reference genomes.
I understand this. Like if you have a very fragmented assembly, how will it work? It will perform. Let's say it would work, but then many of the variants will not be real. I've been showing how with the candidate variant, if you remember, with this, I've been showing how if you have a strain that is very different to the reference genome that you're analyzing, you have this translocation and then a deletion. So I guess you would have the same problem. So the whole downstream analysis part would be a bit problem. So technically it will work, but the interpretation of the variants would be a bit harder.
I mean, you depend on the input data. I guess all this, that's for me, I guess all this is important for the structural variants, but you can always use for, point mutations like SNPs. They will perform equally well with single, but yeah, there will be no point in maybe detecting structural variants if the genome is very fragmented or if you don't have pair reads.
Yeah, you'll be missing many.
Yeah, exactly. So, well, I think that's all the questions I see, and I would like to thank you in the name of ERGA and of all the audience we had. We had over 80 people in the stream session, so I think we had a full house distributed all over the world. So thank you for this clarifying presentation, and I hope people is now downloading PerSVade and trying it. And,
right away.
And maybe we get now an avalanche of questions in the GitHub, and that's going to be a good sign. Okay, so thank you, Mickey.
Thank you
everyone
for listening, everyone.
Yeah, this session. Bye-bye.
Bye.